So I'm Natalie, I'm CEO at Abterra Bio. We're a San Diego-based biotech company, and we do a lot of llama VHH discovery for a variety of different projects. And so I'm excited to introduce you to our technology, Alicanto, and how we're using it to discover diverse llama VHHs using a method called clonal hit expansion. So you may not have heard of our company. We spun out from the University of California, San Diego Computer Science Department, and our team spun out with technologies and expertise in really two main areas. So for the past 15 years, we've been working on methods around computational mass spectrometry and using those tools to do better antibody sequencing at the protein level. And for the past decade, we've been commercializing that as a service called Valence, where we sequence monoclonal antibodies directly from the protein using mass spec. But over the past, I would say, 7 years, we've also been doing a lot of work in immune repertoire analysis. And our co-founder, Pavel Pevsner, and our CSO, Stefano Bonassoni, developed methods for doing accurate repertoire construction from next-gen sequencing reads. They developed a colored De Bruin graph for doing VDJ labeling. And we've rolled that into our flexible service for immune repertoire sequencing and analysis called repertoire. So we sequence everything from hybridomas to single B cells to B cells and tissue, and to phage and yeast libraries. But the technology I want to talk about today is Alicanto, and it marries actually our two core competencies in both proteomics and transcriptomics to create a map of the immune response to do better antibody discovery with a focus on serum. So of course, the past decade of immunosequencing has exploded, and it's taught us that the immune response to challenge is fast and complex and dynamic. Yesterday somebody put up a huge exponential that showed, there's more antibody sequences possible than there are atoms in the universe, and kind of how do we even make sense of that? Any individual at a given time has millions of potential antibody sequences present in them, and wouldn't it be fantastic to have a map to say, this particular antibody binds to this target, and then other attributes that you care about, like affinity or even developability characteristics. So that's what Alicanto helps us do. It looks at that vast immune response and helps focus in on antibodies that have a reactivity profile that you care about, looking directly at the in vivo optimized secreted antibodies present in serum. And so it lets us look at a big tree like this and say, these are the ones that have been selected by the body to be secreted, have high affinity, and have the reactivity that we care about. And it's actually a really efficient way to filter the immune response, because when we look at kind of the B cell receptor repertoire, only about 1 in 10,000 of those sequences, or even fewer, are actually selected to be expressed in serum. So serum's kind of a weird way to look at things. We've developed a lot of methods to do discovery using B cells. So why does serum make sense at all? It sounds sort of complicated. And the main reason is that antibodies are abundant and accessible. So when we work in large animals like llamas, alpacas, and humans, we don't get access to B cell rich tissues like spleen and bone marrow or lymph node. So what we have access to is blood. And if you look at the components of blood, there's 11 orders of magnitude more antibodies in blood than there are B cells. I know we've seen a lot of really big exponents, but that's kind of the difference between grains of sand in a beach bucket and grains of sand in all the beaches in all the world. So what that means is that from kind of a very small volume of serum, there's tons of antibodies. And so we can screen potentially millions of antibodies from not a ton of blood from an animal that weighs 300 pounds. Well, I guess that's a pretty big llama, to be honest. But so from 30 mils of llama serum, there's millions of detectable antibodies that we can be screening. And of course, when we're looking at human, there's obvious reasons to be looking at in vivo optimized secreted antibodies that are detectable in serum. And then kind of from a philosophical standpoint, we take a sequence everything approach. So as opposed to screening hits and then sequencing at the end of the day, we sequence everything that we encounter so that we understand better the context in which those antibodies arose. And that gives us two big benefits. And the first one is diversity. So we know that we're accessing the diversity that the animal produced and delivering hits that are, not oversampling any particular family, but it also allows us to do hit expansion, which is kind of the focus of my talk in the next few slides. So, when you start looking at the immune repertoire, we know that they're not kind of randomly spread across antibody sequence space. They're observed in families, and a lot of those families are really expanded. So what I'm showing here is just from a project of ours where we're doing just a heat map of edit distances between CDR3s that we detected in serum, and we see that there are patches of CDR3s that are very similar. And these show up as these dark blocks down here. On the diagonal, we just have edit distance 0, so that's identity. So every other block here is just a very similar CDR3 sequence. And so if you're sampling randomly from this set of B cells, then you're going to be oversampling these two particular families and not really gaining a ton from it. And you might actually be missing these really unique, rare clonal families that are present here. So by sequencing everything, we can just verify that we're actually accessing the diversity that's present. So, this is old news to you guys, but today I was having a discussion with my barista, and he was very excited about the fact that camelids make unique antibodies. So, llamas, alpacas, dromedaries, and camels have an interesting biology in that they make conventional antibodies that have a heavy chain and a light chain. Those are expressed as IgG1 isotype. But then by some mistake of evolution, they also make these heavy chain only antibodies that are devoid of light chains. And these are expressed as IgG2b and 2c isotype. And of course, the interesting thing about heavy chain only antibodies is that you can pull out that variable region, or VHH. It's a single gene. It's very stable, very easy to express, and we can daisy chain them in ways that make another molecule that's multivalent or multispecific, as we've heard a lot about this week. So just to kind of give a little bit of perspective on what we've kind of come to understand about the llama repertoire. So we've sequenced dozens of llamas and alpacas at this point, and so we just have some observations about what the repertoire looks like compared to other animals that people are working with. So one kind of interesting thing is that you would think that the heavy chain only antibodies are handicapped a little bit in terms of accessing diversity because they don't have the benefit of the light chain rearrangement to give any sort of extra diversity. And then kind of compounded on top of that, we actually observed that the VHH repertoire is dominated by a small number of germline V genes. So not only does it not have a light chain, but they're also all starting from kind of the same germline rearrangement, or the same germline V gene for the majority of the variable region. So here what I'm just showing is a histogram of germline gene usage. So the x-axis is just germline genes, and the y-axis is frequency, and we see that above 70% of the antibodies in the repertoire use the same germline gene. So it's kind of an interesting handicap that an already handicapped molecule adopted. But there are a couple ways that llamas, of course, have found to introduce more diversity. One of them is just by having higher mutational load. So we did a comparison across a few different species looking at mutation load per antibody. And this is just a histogram of mutations at the amino acid level and their frequency in the repertoire across mouse in blue, rabbit in red, the llama is in yellow, and then the human is in green. And you can see the llama, on average, it tends to have a lot more mutations in it compared to the other species. The other way that, of course, llamas generate diversity is at the junction region. So we did the comparison across different species of CDR3 lengths, and we also see that llama antibodies tend to have slightly longer CDR3, although it's not hugely significant. So then in terms of the Alicanto antibody discovery workflow, so we actually started developing Alicanto in rabbit, and we've done projects in a variety of species, including human, but we just sort of fell in love with the fluffy llamas and alpacas. And in our process, we focus on the antibodies that are present in serum, but we don't ignore anything. So we generate data from the immune response, looking both at the proteome using mass spectrometry, and then also at the B-cell receptor repertoire using next-gen sequencing. And our process proceeds through 4 kind of high-level stages. So we're a little bit old school maybe in this audience, but we immunize llamas and alpacas. Then we enrich for antibodies that have the reactivity that we want against our target. We use mass spectrometry and next-gen sequencing to create a large in silico library of that immune response. And then we've built our own software platform to basically in silico pan that in silico library and pull out hits that are diverse and have reactivity to the target. So just looking then at the top track of our process. So when we immunize a llama or alpaca, we're really only interested in the heavy chain only antibodies. So the first step is always doing the heavy chain only enrichment using a protein A, protein G strategy. And then we do antigen-specific purification to pull out the fraction that binds to our target. And then that enriched polyclonal is subjected to LC-MS-MS analysis. This antigen-specific purification is really the part that's customized for every target. And so here we're just showing a simple enrichment against one target, but we often do these in series looking at human and mouse orthologs. So we get species cross-reactive. We do positive and negative selection to get binders to one member of a family and not other members of a family. And we also do competition purification. So we can find antibodies that block or don't block a particular natural ligand's binding site. But what's really nice about having a large animal and being able to get lots of serum is that we can kind of split this and parallelize our proteomic analysis and look at kind of different targets in the same experiment. So in this case, I just want to kind of take a sidetrack into a project we did where we immunized with a multi-domain protein, and then we pulled out antibodies to the whole antigen, antibodies to just domain 1, and then antibodies to just domain 2, and kind of look at, what we get out of that. So in total, we found over 400 different clones with different CDR3 sequences, and we represented those sequences in this reactivity plot. So every column here is one of those proteomic fractions that I mentioned. We immunized 2 alpacas, so we can compare a little bit across animal as well. So here in the first column, we have alpaca 1 purified against the whole antigen, alpaca 2 purified against the whole antigen, and then alpaca 2, we did the domain 1 and domain 2 specific purifications. And then every row in this table is a different clone or CDR3 sequence, and we color it in if we observe that clone in that particular fraction. So kind of one thing to note here is that when we compare alpaca 1 to alpaca 2, we don't see any overlap in sequences. This is not atypical. They're outbred species. And so we don't observe a lot of kind of public sequences across llamas and alpacas. The other interesting thing is that a lot of the CDR3s and clones that we found in the whole antigen purification, we also identified them in a domain-specific purification. So all of these guys here also showed up in domain 1, and so we have an idea of where they bind in terms of their epitope. Same with domain 2. And then we have a set of sequences here that showed up everywhere, and we know they're sticky and they're to be avoided. Kind of interestingly, we found a whole bunch of antibodies that didn't show up again in domain 1 or domain 2 specific purification. And we've since verified that those bind to actually cross-domain epitopes. So you wouldn't necessarily expect them to come out in any domain-specific purification. But then kind of the third interesting observation is that by doing domain-specific purification, we dug quite a bit deeper into the serum repertoire. So for just domain 1, we found a whole bunch of guys that we found the first time, like 100 different guys, but then by doing that domain-specific purification, we found 100 new clones that we didn't find in the whole antigen purification. And so to us, this says that there's quite a bit of depth in the serum that can be accessed and diversity to be accessed, and we just need to be able to kind of understand our targets and our epitope spaces really well to be able to pull out the fractions that we care about. So then back to the overview. During the immunization, we're collecting PBMCs throughout the immunization, and it's a little bit of a misnomer to say that we enrich on the B cell side. We don't actually enrich for any specific population, but we've designed our immunization to take PBMCs at time points that are enriched for B cell receptors that are then expressed in serum. So we have good concordance between the B cell receptor repertoire and the serum antibodies that we're doing mass spec on. With those, we do kind of standard repertoire analysis. We have VHH-specific amplification, we sequence on the MiSeq, and we build our large in silico library of NGS sequences. So in this particular project, we immunized with the extracellular domain of a cell surface marker. We ran our process. We discovered 250,000 unique VHH sequences and 148,000 unique CDR3 sequences. But then kind of where do we go from here? That's more than we could possibly test. So we do the natural thing, which is, organize them into CDR3 trees. But now we've got 148,000 trees. So where do we go from here? So the beautiful thing about Alicanto is that it helps us hone in on specific guys. So we did that affinity purification on the serum. We detected some of those antibodies that passed our affinity chromatography criteria, and now we can overlay them on top of the repertoire. And because now this is our hit, we can then zoom in on a particular set of VHH sequences and explore kind of the characteristics of those antibodies. So that's what we did. So we're just zooming in on kind of the clonal lineage around the hit that Alicanto discovered. In this particular case, we found 87 other sequences that have the same CDR3, but differences in other places. Everybody kind of has their own way, I think, of constructing trees and representing sequences. Ours is, anchored at the germline. So this is the original VDJ rearrangement, no mutations. We make decisions about which mutations happened early in the evolution of the antibody and which ones happened late. So all of the observed sequences from the repertoire are down here at the bottom in these blue squares. But then in our tree, we have a path from that particular observed antibody back to the germline. And so each of these kind of individual little dots in the tree is a hypothetical internal node, some of which actually end up being interesting antibodies, but they're hypothetical and they're not observed in the repertoire. Similarly, we pretty much never see the germline llama sequences in the repertoire. So then kind of an interesting thing to do is say, well, where does our particular hit lie on the tree? And it's actually kind of, there's quite a bit of branching in the tree. And so we were kind of like, well, what's going on? So we just selected a few VHHs across the tree to express. And we found these 6 other binders that kind of have the same binding or higher binding than our original hit. And then we also found a number of guys that for some reason or another, they took a wrong turn. They took on mutations or changes that ended up negatively impacting binding. So if this sounds like you're having deja vu, that's because this kind of within clone hit expansion was talked about last PEGS by our CSO Stefano Bonassoni, who did a nice job of exploring a clonal tree and then actually associating the affinity that we find on the tree with a multiple sequence alignment and giving an idea about mutations that were pretty essential to be conserved to maintain binding to the target. And then also a lot of ideas about other mutations that were either neutral or negative to binding to the target. And if you want to catch this talk and you didn't, Abterra Bio has a YouTube channel, and you can catch Stefano talking about this particular kind of hit expansion on the interwebs. So in these particular hit expansion case studies, we're always looking at these really nice trees that have lots of different candidates. There's many options to choose, but more than half the time you end up with an anemic tree that looks like this. So what do you do if you actually find a rare clone? There's nothing else that's similar to it. There's maybe 1 or 2 other guys that have a similar CDR3 sequence. And so what kind of options do you have to generate more hits from your repertoire? So that's the idea behind clonal hit expansion, is to jump from one CDR3 family to another CDR3 family, looking for a retention of the specificity and hopefully lots of other candidates to try out for the other properties. So just pictorially, this is what we're looking for, is this little anemic tree, has a cousin in Wichita that has just tons of buddies. So kind of the way we find similar CDR3s is to take a zoom out from the clonal lineages that I showed previously, where everybody has the same CDR3. We just, zoom out, and now we're looking at CDR3 space, and you can find sequence similar CDR3s just looking in the repertoire. So here's our particular CDR3 sequence, and we just built a network of other CDR3 sequences. And in this case, we draw an edge between CDR3s if they're edit distance 1 away. And so there are other candidates. When you align them, they tend to look very similar because we're only looking at edit distance 1. And so when we look at things sequence-wise, as other people pointed out, it's a little bit simplistic. Where do you draw the threshold? Many things that are actually kind of way out here on the tree are actually edit distance 4 away, and if we start including all those guys, we're going to have tons of noise. We know that not all mutations are necessarily created equal, and so we want to represent that somehow when we're trying to do clonal hit expansion. So what we elected to do instead was to look at the physicochemical properties of the CDR3s. So we built an autoencoder that maps from CDR3 amino acid space to an embedding in a physicochemical space, and that allows us to identify CDR3s that are proximal in that physicochemical space, but maybe really different in terms of their sequence. And we look at a number of different properties, and we've trained a number of different models. And so for our particular CDR3, we found a couple of different embeddings that actually were very similar in physicochemical space, but quite different in terms of sequence space. So one of the binders ended up being kind of, just as good as our hit, but not necessarily anything to write home about. But one of them had kind of an interesting story. So from our particular orange clone, using that as the seed in the autoencoder, we found this blue seed that was in the repertoire. It's rare, so there were only 3 different VHHs that had that CDR3 sequence in the repertoire. It's distinct, so it's only about 60% identical to our original CDR3, so not something that we would necessarily consider. But then in terms of binding, it ended up being our second best binder in the whole project. So we're really lucky to find it. Now, the goal of the autoencoder is not to go from big trees and find these kind of expeditions to find little small trees. In this case, we were biased because we were looking at big trees to be able to do the within clone hit expansion. But the purpose of the autoencoder is to be able to go from these rare blue guys to the big orange guys. So, you can imagine a separate universe where you found the blue guy. It had 3 relatives. All of them have maybe some N-glycan site that you really want to avoid. So you use the autoencoder, you find the big orange guy that has 90 different buddies, tons of very similar CDR3 sequences that you can then explore kind of the natural space of related antibodies. The other cool thing is that in this case, the blue or the orange sequence were both from the repertoire, so they were observed sequences, but the CDR3 actually could be seeded with anything. So a lot of times when we analyze the serum, we identify CDR3s that aren't present in the repertoire. We can use that CDR3 from serum as a seed to then find a closest relative in the repertoire, and that has actually been fruitful for finding these rare serum-only clones. So then just to summarize, so Alicanto is our technology for doing VHH discovery. It allows us to screen millions of antibodies and map certain properties onto the vast immune response. And we've incorporated into Alicanto 2 different types of hit expansion. So the first one being the within clone hit expansion that Stefano talked about last year, and now this new autoencoder enabled clonal hit expansion that lets us find completely new CDR3s that are related to our hit. So then I would just like to thank you so much for sticking around for the last day. Our whole team at Abterra Biosciences, and particularly Tiago Lima, who's here this week, and hopefully you had a chance to meet. Part of Alicanto started out as an NIH project, so we're grateful to the NIH for the SBIR funding. And then, of course, the adorable llamas and alpacas that are so gracious and contribute immeasurably to the project. And I'm happy to take any questions.